everyone. Thank you so much for joining us on this St. Patrick's Day, and welcome to today's webinar, focused on getting to know us a little better. My name is Kristen Ferrante, and I will be your moderator for today's webinar. Presenting is Dr. Weston Umstead, Manager of Technology here at Chiral Technologies, and Konstantin Haralampopoulos, our Director of Business Development. Now, I would like to introduce to you Dr. Weston Umstead.
Good morning and good afternoon to all of you. Thank you very much for joining us today on this St. Patrick's Day for our one-of-a-kind or first-of-a-kind Getting to Know Us webinar. Most of you who've tuned in today are probably familiar with the chiral core business of Chiral Technologies, whether it be chiral columns or chiral separations, but we also have some new services that we are offering with our affiliates in Daicel Chiral Technologies India that Konstantin will be talking about. As a brief overview of those topics, I'm going to tackle the first 3 bullet points here, which are all related to the chiral core business, and then Konstantin's going to jump in on the final 2 points, certainly not the least 2 points, to talk about those new services, including impurity isolation, extractables and leachables, and stability testing. So we have a number of Daicel Chiral Technologies affiliates that are located around the globe. Here in the US, we're located in Westchester, Pennsylvania, but we also have colleagues that are in Strasbourg, France, CTE, Hyderabad, India, Shanghai, China, and in Tokyo in Japan. And we are all a part of the global Daicel network who are responsible for the chiral core business, the chiral separations piece of what Daicel does. So we offer a number of services here in the US, same as around the globe, for analytical and preparative chiral method development. One of the questions that we get asked most frequently is that I have a chiral molecule, what column should I use to separate the enantiomers? The reality is the answer to that question is quite complicated. There are a number of interactions that take place on column that are not easy to predict, whether it be between the mobile phase and the analytes, or the analyte and the chiral stationary phase, which makes it extremely challenging for us to just predict based on structure that IG, for instance, would be the best column, or IBN would be the best column. And the other reality is that chiral columns are expensive when compared to other generic achiral phases. So we certainly wouldn't want to recommend a column that we don't know for sure will work, knowing that it is an investment for you as the user to purchase this column for a given application. So the best approach is to screen the compound on our library of chiral stationary phases, which I might refer to in the upcoming slides as CSPs. And we screen those CSPs across what we have found to be a proven set of mobile phase combinations to yield good baseline separations for both analytical and preparative applications. So for analytical chiral method development, we offer a free screening service here at CTI. For analytical method development specifically, we will provide you with a report which demonstrates a separation on our columns, which includes the column recommendation, the mobile phase that we found the best separation on, and then other important method conditions like temperature and dilution of the sample. This is just sort of a snapshot of what that report looks like, pieces of that report. This was a separation that we developed recently for nicotine. You can see over here on the left-hand side the chromatograms for the racemic, the separation of the racemic, and then in this case we had an analytical standard for the minus enantiomer so that we were able to do elution order determination. So you'll be provided with the chromatogram, and then you'll also be provided with a table that looks something like this, where you've got the column, the mobile phase conditions, flow rate detection, and so forth, for you to be able to essentially duplicate this method in your own laboratory. Generally for analytical chiral method development, we're screening on smaller particle sizes like 3 micron or sub 2 micron. I say typically because there are a number of applications on 5 micron. 5 micron is sort of interesting in that it can straddle the line between preparative and analytical. So if you request it, we can do 5 micron, but know that typically we will be screening as a default on 3 micron or sub 2 micron unless we're told otherwise. No limitations in this case. For mobile phases, we have the ability to screen normal phase, reversed phase, polar organic mode, supercritical fluid chromatography with SFC, as well as developing mass spec compatible methods for you. And we do perform some minor optimization. So if we do a screening and we find a partial separation, something maybe that's 75% baseline, we will do that optimization to get it baseline and provide you with a baseline separation. But we do also offer a 4-phi in-depth method optimization, which I won't really touch on too much here. It relates a lot to what Constantine will cover, but just know that we can cover a full suite of services for you. For preparative chiral method development, it's fairly similar. We do provide, again, a free service where we will perform a screening to identify a candidate method for scale-up and then assess the productivity and the stability of that method to give you a sense or determine the potential scale. Now, this service is offered for both in-house preparative separations that we will be running here in the US, as well as outsourced preparative separations that we would pass off to our colleagues in China or India as an example. But we also offer this as a service for assisting in the purchase of a prep column. If you have a system in your facility that's capable of handling a 2, 3, or 5 centimeter prep column, we can do the work for you in developing the method to help assist in you purchasing that prep column. This is a report that we don't share openly as a part of the in-house separations, but would be provided to you along with a column sale, where we do the assessment for the productivity. The productivity is based on the solubility of the material, the cycle time, and then the maximum injection volume on that method. And this is on an analytical column, so 4.6 by 250. You assess the productivity, and then that can scale proportional to the inner diameter of the prep column that you're choosing. And of course, we would walk you through that whole process to give you an idea as to how much material you could theoretically process with your column that you'd be purchasing. For preparative chiral method development, the screening, as I mentioned, would be on the larger particle sizes, 5 micron or 20 micron, depending upon the ultimate application. We do limit our mobile phases in this case because solubility is extremely important. If we go back to that worksheet, solubility and the amount that we can load on the column is a huge driver in improving productivity. So we generally stay away from low solubility mobile phases. We also stay away from reversed phase if it's an in-house preparative separation, as we don't have the large scale lyophilization capabilities to be able to remove the water. And of course, we do a full optimization to ensure that we're providing you with the highest productivity method that we can possibly find. Here in the US, we've actually built a new production facility, which is located out in Ann Arbor, Michigan. We have the 2 facilities now here in Westchester, Pennsylvania, and then out in Ann Arbor, Michigan, where we are able to handle milligram to multi-kilogram HPLC separations. We are your local separation solution. Anything that you want to do here in the US, we can certainly handle it, or we can work with our colleagues abroad to be able to do that. We pride ourselves on being fast, reliable, and flexible with whatever your situation might be. So even if you think it might not fit our scope, we would love to be able to talk to you about those projects and try to figure out a good solution for you. A lot of the things that we see end up going from just perhaps a screening to large scale separations, whether it be in-house or globally. So the analytical method development and the preparative method development aren't always separate sorts of services. A lot of times they end up kind of melding together as one. Modafinil is such a case that we saw in 2003. It was licensed to Cephalon as a wakefulness promoting drug. And it was found at that time that the R enantiomer happened to be longer lasting than the racemic, which isn't always the case in the pharmaceutical industry, but oftentimes is, where one enantiomer is more active or more beneficial than the other. So we performed an initial screening to find a separation, identify a separation to be able to separate the racemic, which we were able to do successfully. And we were able to scale that up to an initial 120 kilogram project, which provided 60 kilograms of the desired R modafinil for phase 1 clinical trials. Further scale up then via simulated moving bed, as I have abbreviated here, SMB, which is essentially a very large scale, high throughput sort of HPLC technique. That's a continuous process allowing for very large throughput of material. But scale up via SMB provided 600 kilograms then of material for phase 2 and phase 3 trials. Now, ultimately, most pharmaceutical companies are going to spend the time to try to develop an achiral synthesis to avoid chromatography, because in the end, you are going to be losing at least half of your material. That's the undesired. But in this case, the take-home message was that chiral chromatography provided a pathway for Cephalon to be able to move through phase 1, 2, and 3 trials before the initial patent expired in under 24 months, while they were also spending the time to develop an achiral synthesis. So they were able to move through all of these clinical trials while sort of working towards the ultimate development of an achiral synthesis. So time can be saved, money can be saved by implementing a chromatographic process in the interim, even if it's not the ultimate long-term goal. A few other examples, we're currently working on a proprietary compound. It's flavonoid based. Unfortunately, I can't share the structure with you, but know that it's a drug with multiple indications, one of which is acute myeloid leukemia. We screened it at an earlier synthetic intermediate under non-GMP conditions here in the US, and we were able to provide the company with 7 batches in total of 100 plus kilograms of material, which ultimately went on to support their further GMP synthetic steps. And I believe that compound is currently in phase 2 or phase 3 clinical trials. And then lastly, another one that we're currently working on, again, unfortunately, I can't share with you the structure, but it is a pair of Atrip Isomers, so it's a little unique. It's not the classical sort of one chiral center. We screened and we developed an SMB purification method, which was somewhere in the neighborhood of 3.4 to 5 kilograms of enantiomer per kilogram of CSP per day. The productivity in this case looks different from what I showed you before in terms of milligrams per hour or grams per hour, because SMB systems are, the productivity on SMB systems is based on the size of the column that's being used. And so a 5 centimeter system, for instance, will use 300 grams of CSP. This productivity then is easy to adjust to whatever the SMB size is. So it's looks a little different, but it's essentially the same thing. How much material can you do on the system in a day? The results of that simulation were good, but there was a little bit of uncertainty. And so what we're currently doing is running a 500 gram benchtop SMB demo to prove those conditions out before scale up, because obviously committing to a large scale up with any sort of degree of uncertainty is not as good as moving forward with complete certainty. So we're working on that one right now. For preparative chiral method development, in addition to those services I just mentioned, we can develop HPLC and SFC separations, which can transfer to our global Daicel network. We have capabilities globally that can handle all sorts of different projects. And as I mentioned, we can do SMB separations, we can do the loading, and we can also do the simulation of those conditions to determine their productivity, which can then either be run by our global partners or transferred to another site for running. I'll show you the map again. I kind of pointed this out at the beginning that we're all a part of the Daicel network, the Daicel Chiral Global Network. Globally, we have the separations capacity in the US, in Japan, France, China, and India, where we can handle milligram to 100 plus kilogram HPLC, SFC, and or SMB separations. We have redundancies in capabilities and systems. So if you have a project that, say, is 200 plus kilograms and you need it done in half the time, we have multiple systems which can handle that sort of work to meet the deadline or meet the timeline that you're expecting. We can do those small scale SMB demos to prove out simulation conditions, and we can also do small scale HPLC and SFC demos for the same reason, just to ensure that the conditions scale appropriately before committing to a larger scale project. And of course, we have several sites that are fully cGMP compliant. We are all Daicel. I mentioned it just a few slides ago. Although we are located in different places, we are all owned and operated by Daicel, which means that we hold ourselves to the highest of Japanese quality standards. We have a seamless technology transfer between all of the sites. We work with each other on a regular basis. And because we are all the same company, there's no sort of issues in terms of interpreting data or making sure that the capacity fits or something like that. And the other important thing is we never work with any outside or third-party vendors. So we're not sharing any sort of information with any external CROs or CMOs. Everything that we do in-house, and we keep it protected. Even information that is shared amongst the employees is only shared on an as need to know basis. So we have a high quality standard making sure that your projects are protected and safe. So what else is available? I mentioned a number of services related to the chiral core business, and we have a lot of other things that we are now also offering at our affiliate at Daicel Chiral Technologies in India. And so for that, I'm going to pass the presentation over to Constantine, who will discuss those new services. So Constantine, take it away.
Hello, Wes, and thank you so much, Kristen and Wes, for the nice introduction and your portion of the presentation. And to everyone out there, thank you for attending our webinar. I know from what I saw on the sidebar, there are a huge number of attendees from all over the world. A big thank you to all of you across the various time zones, which may not be convenient, but it tells me one thing, that life science companies out there are looking for dependable value for money testing. And this is what we deliver at Daicel Chiral Technologies India, which is the acronym DCTI. And with that, I just also wanted to give you a little bit of a disclaimer here, given the time constraints. For my portion of this presentation, I'm giving an overview of the analytical service solutions and not from a technical nature, digging deeper. Just the same though, don't worry, I'm happy and ready to continue this dialogue confidentially and learn more about your team's testing needs. In addition, as a side note, we also, as DCTI, generate a monthly industry newsletter, which is focused and informational and educational in nature, in which our subscribers find value and benefit on the latest analytical testing techniques. And also, we do exhibit at all major life science trade shows and conduct face-to-face meetings. Obviously, with COVID being in our radar screen, we do intend on participating during the second half of 2021 to these trade shows. So keep an eye and ear out for us. I'm happy to engage further for that type of deeper dive. And with that, I would like to then proceed with the presentation at hand. As Wes said, we are all Daicel. We work with companies such as yours in attendance. Some of you know us as being a solid performer in any given solution service that we provide. To those of you out there that know us already, this presentation is also to expand your knowledge base as far as what we can provide further to you. To newcomers here, again, welcome. We hope that you find this portion of the presentation informative and engaging, and think of us for your next testing needs. Now, as Wes had alluded to, our network is expansive and global. Daicel is a large Japanese-owned company, but however, we work on a very roll-up-your-sleeves, can-do attitude basis where we take our clients' problems, issues, and testing solutions very seriously with fast responsive times. Our physical location for Daicel Chiral Technologies India is indeed in India in Hyderabad, which is our analytical center of excellence site. As you can see from the photo here, this is a state-of-the-art facility, which is equipped, as we say, with the 3 E's, our expertise, state-of-the-art equipment, and experience with our staff. And again, this is a photo here of that wonderful facility. GMP services, as Wes also alluded to, we are a CGMP and ISO standard facility and proud of that. We are accredited by all the global regulatory agencies, as you see the sample bullet points here. And our inspections and audits have passed with flying colors. So there's no issue of red flags or observations. We cater to the global 100 life science companies for their testing needs, and they trust us for that, and we are proud of that fact. Now, let's get into the actual offerings that we have. We'll start with the one in which we receive numerous inquiries and also orders on an everyday basis. It may not be the sexiest offering, but I will tell you it is the one that has brought us a lot of open doors in order to prove ourselves going further. And these are our Pharma Standards from as low as the milligram up to the multi-gram quantity. And what is our secret here? Competitive pricing. We have a robust catalog of such standards of assured quality. We strive for 95% purity, and which is, by the way, accompanied by an FDA compliance certificate of analysis. Now, related to this is the custom impurities offerings. As seen in this graph, and forgive me, there's a lot going on here, but we're happy to share details with you in expanded presentations that we have for each of these offerings. We do a deeper dive for any type of testing that you need, whether it be impurity testing, whether it be extractables or leachables that I'm going to showcase a little bit later, microbiology and so forth. So I just want to let you know that I'm really just giving an overview here, but happy to engage further and deeper for any of these solutions. So related to impurity standards, we basically have our own methodology step by step. To summarize, we start with your specific requirements. We enter with a feasibility phase, move on to characterization, which includes the structural elucidation, then move on to the isolation of the target quantity through preparative HPLC, which then brings us to the synthesis that occurs through route design. And in the end, we have the target impurity at hand with a COA from, again, FDA compliance standpoint. Now, method development validation is the really the claim to fame that we have with Wes's group. And what I mean by that is, even though this slide does say method development and validation, we also undertake optimization and method transfer as well. Many times, more than not, we perform complementary initial screening for our clients. And with that, we work hand in hand with many of Wes's clients on this type of advanced type of method validation. Mind you, we also have upcoming case study in which this will also be sent to our recipients here today in our subscriber list of a case study in which we conducted and performed recently, which will give you a little more real life history on a sample such development and validation. Stability studies. This is also a bread and butter study, which obviously we have seen growth. And by that, I mean, we have expanded our facility in Hyderabad to an additional 80 square meters of stability chambers, because of the increased demand. It goes without saying these chambers have 24/7 backup power supply redundancies to basically ensure the consistent and continuous temperature relative humidity settings that our customers require for their studies. These studies also have SOP, standard operating procedures, and documentations in place for the loading and withdrawal of the samples as dictated by the client's requirements. From what I see here on the chart, what really rings a bell, I see a lot of photo stability that we undertake and formulation development as far as stability goes. Now, extractables and leachables is really one of the two growth areas that we see at DCTI, and really for good reason. This, as you most likely know, is an increasing and stringent regulation out there concerning the potential leaching of packaging materials into the drug in question. So obviously patient safety is the utmost concern here. So we know that new and innovative compounds are always coming out, and with that, these are being used to house or deliver medicines, and thus need to be strictly tested. And that goes also for rubber stoppers, seals, tubing, and so forth. It applies not only to pharma medical device, but also to the food and cosmetic industries, among others. Our basic radar screen now has seen the 4 eyes that I show up here as far as inhalants, injectables, infusions, and implantables as being a big bulk of our business currently, but not only. Dermal and topical applications also play an important place as far as volume of work, as does from a vision care and nasal spray standpoint. This slide really probably is the most technical slide that I'm going to get into today. It's really just showcasing the equipment that we use for each class of compound within E&L. And as you see, DCTI has the appropriate instrumentation for accurate E&L testing based on the compounds at hand. And again, we have a dedicated presentation of around 30 minutes that digs deeper in this type of testing solution if and when you need it. What I could say also is that it's very critical that E&L testing be done right the first time based on its relatively higher cost of study and the longer, relatively speaking, again, time frame to conduct such a study. So if any one of you out there know you have a product that's going to be launched, you cannot afford just extending the budgetary requirements for earmarking for E&L if you did not do it right the first time, which would also prolong its launch to the marketplace. So very critical. Microbiology testing. Although it's not a growth area per se, it is a sizable amount of our business, and any lab, as you know, out there worth their weight needs to have a solid and dedicated microbiology lab. And we are proud to have the breadth and scope of various, almost all-inclusive microbiology testing in-house there at Hyderabad, ranging from sterility and all the way down to the latest ones that we've seen many times, expanded further. Preservative efficacy testing comes to mind, as well as bioburden testing is another one that we've seen a lot of business with, especially with medical device companies. Non-routine is really a misnomer. It's not non-routine because of the non-frequency. It's non-routine because it requires a little more expertise in here. And again, we pride ourselves as far as completing the whole continuum of microbiology testing in-house. And here again, I show the bioburden type of testing. Endotoxin is another one that is widely requested, as well as stability testing from a microbiology standpoint. Now, the other growth area that I wanted to mention, aside from extractables and leachables, is within the large molecule domain. And I'm specifically speaking biopharmaceutical services. From this graph, the spectrum that you see, we have been very proud in the last year or two to complete this puzzle of providing a one-stop shop in all these types of biopharmaceutical services. And the reason and the driver is, well, it's now the future and growing exponentially. The analogy that parallels this is the generic medications that were brought to the marketplace, say, I don't know, 35 to 40 years ago. Biosimilars are in this position currently. So DCTI has recognized this from years before, jumped ahead of the curve, invested steadily in this type of testing in the last 10 years, and we are fulfilling the most requested services in this sector, as outlined here, but not only. Peptide Services. This is another area related to biopharmaceutical in which we basically have seen what was requested, voice of consumer out there in the marketplace, and aligned our expertise accordingly in order to support such testing such as peptide synthesis, generic peptide APIs, peptide mapping. There are other services that we provide that are not listed here. Of note, if I remember correctly, they could anything from process development and tech transfer, amino acid analysis, again, goes hand in hand within the space of biopharmaceutical testing. And lastly, within, but not least, is in vitro bioequivalence studies. In this particular space, what I have seen and the team has been focused on is dissolution testing specifically, and not only. We've seen our share of the other three here listed, but I'm proud to say within dissolution studies, we do have the various apparatus from 1 to 4 in order to conduct your specific testing needs. Lastly, to sum it all up, what does it really mean to you? You're here on this webinar and you're saying, okay, so what? And the way I'm going to answer so what is give us a chance in order to prove to you, as we have done to the attendees here today, in which we've earned their trust day by day by providing integrated services. That means working hand in hand with people such as Wes for a seamless integrated service solution and delivery. A global network where we have dual, sometimes even triple sourcing and backup methodologies and contingency plans in order. Quick turnaround time. And when I say quick turnaround time, I'm not only speaking about the actual testing itself, but also the quoting process, which I know from other laboratories can be a nightmare, and also from a reporting standpoint. Because again, we perform in a very nimble, roll up the sleeves mentality here, even though we are part of a larger respected Japanese company. Technical expertise, most of our staff out of Hyderabad either possesses a master's or PhD with 10 years experience at least and very low turnover. Advanced technologies, I just mentioned briefly state-of-the-art equipment that we have. And the seamless communication. Basically, any of you in attendance here today know that I personally, as a face and voice of DCTI, I've been able to provide 24/7 service. And with that, we generally love what we do, and we wish to demonstrate to you to earn your trust and give us the opportunity to serve your team. And that's why we are the right partner. I'll close with this before handing it over to Wes again. I think a question I received from the audience is if E&L is applicable for in vivo and not only for in vitro. The answer is yes. Implantables is just one case scenario, and I'm happy to take this offline with that particular attendee. And I think there were some flatter here, another translation. Can we translate this into other languages? I think this is in the works. I think this, Kristen may and Wes may provide commentary that this presentation is being recorded. With that, I'll give it back to Wes. Thank you for your time here today. And again, I am at your service, as well as Wes, to provide exceed expectations in our roles. Wes?
Thank you very much, Constantine, and thank you to all of you who tuned in today. A few email addresses here for you if you have any questions. relating to the chiral core business, the chiral separations piece, please feel free to reach out to me. And anything related to the analytical services labeled Pharma Standards, please feel free to reach out to Constantine. There is a copy of the handout, the slide presentation. You can certainly download that. But if you'd like something else, we have a number of other webinars that we've done in the past. Please feel free to email one of us and we can certainly put you in contact with somebody who can provide you with that information. So thank you once again for joining in. Please enjoy the rest of your St. Patrick's Day, and we will hopefully be discussing these projects with you in more detail in the near future.